This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 734, recorded on March 19th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's here in Western Mass. It's clear skies, 36 Fahrenheit, 2 Celsius, and blustery on the day before the vernal equinox.
Yeah, it's really windy here. We went to the Mouse House this morning, and it was really cold. It's 3 degrees, but it is very sunny here today.
Wait, it's 3 degrees in the Mouse House?
No, here in outside. In the Mouse House, it's always 72. And
okay, yeah. You're going to have some problems there.
No, just like on the International Space Station, it's always 70 degrees and clear. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 63 degrees and sunny, headed for 67. You guys have the wind that blew through here over the last couple of days. It's nice here today.
From Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's 43 and sunny here, and obviously also windy.
And from here at Columbia University, Amy Rosenfeld. Welcome back.
Thank you. I moved the annex. The annex now is my lab space.
Yeah, I said you should go in the lab because when you sit here, it's hard to separate the voices. So, and Amy said you're banishing me, huh?
It's fine.
No, but it's a really good image. You got nice, colorful boxes on the shelves.
Yeah, it's an active lab. I actually do bench work and write papers and
you've got good side lighting from the window.
Side lighting, and nice. She has a biohazard container there, right?
I do. I have several.
It's all good. We have a guest today coming back, I think for the third time, from the University of Pennsylvania, Perelman School of Medicine. Susan Weiss, welcome back.
Hi, I'm here in sunny Marion, Pennsylvania, just outside of Philadelphia. It's 45 degrees, and I don't think it's very windy. I was out earlier. Yeah. And it's a special day. It's my son's 38th birthday.
Wow. Congratulations. Cool. What is your son doing?
Well, he has been a professional magician until recently. And since that's sort of been difficult, he's now a middle school teacher.
Wow. So he could do tricks and he could do magic for the class, right?
He tells them he can make them disappear if they don't. Yeah. Things like that. It's pretty corny.
That's just
How many parents want to do that?
Yeah, make
to enroll in that school.
That's funny. So before COVID, he was getting gigs, right? And
he was completely busy.
Huh. All over the place or just Philadelphia?
All over the place. He was supposed to go to Dallas. He once did, I think it was at Madison Square Garden once. Not the main act, but Washington, a lot of places.
Cool. Yeah. Well, Susan, we had you last here on May of last year. Gosh, time went so quickly. And that was really at the beginning of this whole, I want to call it something that mess. Yeah, thank you, mess. I had another word in mind, but I can't really say it. So I thought we'd get you back and catch up here. Before we start, Rich Condit wanted to make a public service announcement.
Right. I just want to plug a new event or series of events called the COVID-19 Vaccine Education Town Halls. And there's a website for this that's part of the ASV website, asv.org/education. This is the brainchild of a virologist at University of Virginia named Dan Engel, and he's recruited a pool of in excess of 50 experts. These are academics, including Brianne, including Susan, right? These are professors, physicians, mostly with academic credentials who have some insight into COVID and SARS-CoV-2. And it's the whole idea is to help people understand vaccines, in particular people who are vaccine hesitant or have questions. And this is done by supporting a series of brief town hall meetings. They last half an hour to 45 minutes. each hosted by two out of this panel of experts, and they are numerous. They'll launch next week, Monday, the 22nd. And you keep checking on the site, and you'll see, if you look on the front page of the site, there's a button where it says register for town halls. And if you do that, you see a list of these things. It says who the hosts will be and when they will be. They're free. You can register for any one of them. You can register for several of them. If you keep checking back on the website, it will be updated with more as they come along. And we hope that this will provide quality information and a place where people can go to ask their questions and get answers.
Okay. We'll put a link in the show notes. When Rich said Dan Engel, I thought he was talking about a celebrity. So I was Googling left and right. And nobody came up. So
there will be, I should say, there've been a couple of one 30-second teaser and another more 5-minute promotional videos that'll show up on this site eventually as well.
Okay.
They aren't quite ready yet.
All right. Now, Susan, I have a kind of a list of things we could touch on, but I don't know how far we'll get, but let's see. And I wanted to start, we had a discussion I don't know, I have no idea when, but a while ago about hydroxychloroquine and Tempris, and we ended up talking about entry at the surface and from within endosomes. And you had emailed me saying, this is not something new for this virus. So I thought we'd have you tell us a little bit about that. But by the way, I wanted to ask you, are you the longest working coronavirologist at this moment in time? No? Who is?
Second longest, Julian Leibowitz.
Ah, okay. He's in Texas somewhere, right?
Yeah. So interesting anecdote. When I got interested in coronaviruses, if you want to hear this.
Yeah, sure.
I was a postdoc at UCSF, and he was an new assistant professor at UCSD. And when I got interested in coronaviruses, I contacted Ian Kennedy, who you may or may not remember. Mm-hmm. He was a professor at UCSD. He was working on IBV. I told him Mike Bishop told me about him. So I contacted Ian. Ian said, Julian Leibowitz is a young guy just starting to work on coronaviruses. Why don't you talk to him? And so I did. And I went down to UCSD and we worked together for a week or so. Then he came up to UCSF, slept in my backyard in a tent because he was allergic to my cats. And we got a lot of work done in those 2 weeks. And that's how I started. So Julian was like a few years ahead of me, and he's still an active coronavirologist. And I think probably we are most senior, the two of us.
Yeah, I think that's cool.
Yeah. Kay Holmes is retired. She was she's the one that I mentioned. She really did the very first stuff on Spike.
So as a, senior person in the field, are people consulting you as they should often, or not as much as they should?
No, probably too much and too little.
Okay.
I get a lot of emails asking all sorts of questions. A lot of people want to work on MHV now, murine coronavirus. You can believe it. I finally deposited it with BEI because there are two cell types that MHV grows in. One of them is called L2 cells. And there happens to be a line of L2 cells at ATCC that are actually rat cells, and they're not the right cells, but they have the same name. So a lot of people have been thrown off course by that. So I deposited the real mouse cells at BEI.
Do you think the influx of people in the field will continue afterwards?
I don't know. I think some people will stay, but many will probably not, because it probably won't be easy to get funded.
I was going to say, I think it may depend on how much of the influx of money stays in the field.
I don't think that people doing basic science have gotten a huge amount of money out of funding. I don't know.
Yeah. All right.
Well, we did not.
No, we certainly did not. But some people have, right?
But it's all basically to immunology and stuff.
Yeah, I agree.
I got a supplement to my grant, but that's probably because I already had a grant on MERS.
You should, because you're in the field and you know a lot and you have resources. And a lot of people come in and they're actually not even virologists. They just have an idea, which is not a bad thing. But
some people, I think it's really good. They've used new sophisticated techniques applied to old questions. And so that can be really good. I know the clinical immunology is really good.
Yeah. So let's talk about entry. You'd sent this PowerPoint slide, which is from 2013, I think. Coronaviruses use two routes of entry into the cell. So tell us the history of this.
Well, it's older than that. So just in the very beginning, there's always this question where viruses enter through an acid-dependent, low pH-dependent endosome or by the surface. And I think early on, we thought coronaviruses entered at the surface. And then other people started saying, no, they enter through endosomes, that it's sensitive to low pH or to high pH. And then I think what it turned out, at least in the MHV case, that it was both, because there were different strains that used more or less of each pathway, and there were different cells that favored more or less of each pathway. And to sort of think of it more simplistically, I'll just give MHV examples because it's simpler. Most MHVs have cleaved spike proteins, have furin sites and cleaved spike. MHV2, for example, has a completely uncleaved spike. It's completely pathogenic, causes severe hepatitis, but it doesn't have a cleaved spike. And when we and others looked at all these strains, it seems like the strains that cleave well, for example, the very highly adapted brain strain JHM, is completely dependent on surface proteases for entry, goes in completely at the surface. Not through TMPRSS2, because mouse cells didn't have very much TMPRSS2 when we looked through other proteases. And MHV2 at the other extreme that has completely uncleaved spike was totally dependent on cathepsin pathway. So that's very simplistically looking at it. And it really, even cell lines, or even Kay Holmes's original paper in 1985 showed that there were different amounts of cleavage in virus produced by different cell types. Mm-hmm. And so those viruses which had different cleavage states would enter by both routes, but proportional to whether they were cleaved or not, just in a very simplistic way.
Yeah. Now
and just real quick here, for people who didn't see your previous episode, MHV is mouse hepatitis virus, which is a kind of a big standard model in coronavirology, right?
And it's got many, many different strains that makes it even more a really interesting virus to look at. Different strains, different spike proteins. Yeah.
Now, you said one of the, I'm sorry, 1 second, Brianna. One of the viruses does not have to be cleaved at all? The spike doesn't have...
MHV2, when it comes out of the producer cell, is not cleaved at all. And that's true for 2290 and NL63. They're not cleaved. You don't have a site. But clearly, they must be cleaved to go in. So the assumption is that when they go in through the endosome, this is what I'm trying to figure out in my own head too, that cathepsin must be cleaving at both the S1, S2 site, and then there's the second site. You know about the second site, the S2 prime site that opens up the fusion peptide. And I was looking, there's a really good, the person that really knows this stuff well that you should have on is Gary Whitaker and Tom Gallagher. Both of those are really... They've studied this entry and enzyme utilization really, really well. And Gary has a review where he shows how S1, S2 can be cleaved mostly by furin, but sometimes other enzymes depending on the virus, and the same thing for the S2 prime site. And he and Tom have done things like mutating the sites or changing the sites and just seeing what happens. Because not only does it have to enter, but a lot of these viruses do cell-to-cell fusion. So MHV2, for example, you infect cells, you see no fusion at all, and virus comes out uncleaved. If you take those cells and tweak them with trypsin, they fuse like crazy. So putting out an uncleaved spike on the surface as well. But then it's perfectly infectious. That's what's so mysterious. But it really is. But that's true again for the SARS-1, right? Not cleaved.
But that's what I was alluding to earlier about her work with the aspartic acid and histidine, where you change the histidine to the aspartic acid in spike, and it still infects, but it doesn't cause incision. Or maybe I have it reversed.
So this was interesting. We didn't make, again, I don't think we can fool Mother Nature. We didn't make that mutation. What we did was we put virus...
You selected for it, I thought.
We made persistently infected glial cells. So we selected for replication in glial cells. What we got out, and in independent lines, we got out virus with not a lot of mutations, but they all had this H716D. I remember that really well. So they screw up the furin site with an acidic amino acid, and it doesn't cleave. It cleaves very late and fuses very late in infection and very little. That virus was really infectious in the brain, but as it had been adapted to glial cells, but it no longer caused liver infection, which is the other main target. So it lost one tropism, but it kept, but then, right. But MHV2 is highly hepatotropic without a cleavage site. So I think the message is that each one of these viruses somehow adapts to it either needs or doesn't need the cleavage site, but it optimizes its own situation.
So in one of our previous episodes, the question of what happened to the portion of spike that is cleaved off came up. And so do we know anything about what happens to that portion of spike? And perhaps could that portion have some other function that might differ between the viruses that have uncleaved spike and the viruses that have a cleaved spike?
So you mean the part that cleaves off, you mean the S2 prime piece, that most C-terminal part of S2, or you mean the whole of S2 or S1?
I think that anything really, I've actually been wondering in general about general functions of spike in addition to fusion. So
I don't know, but like some of the spikes are really hyper unstable, like this JHM, so JHM was, at least the strain we used was selected by 50 passages in mouse brain. So it's super neurotropic. It completely goes in on the surface, and it's got this really long, very extra long what they call hypervariable region of the spike. And so it does receptor independent spread. That's a whole other story. But the point is that S1, S2 is really unstable, and S1 pops off really easily. Tom Gallagher has a bunch of papers about how each MHV spike has a different level of stability and shedding of S1. So you can actually inactivate the virus. So we sometimes grow JHM at a low pH because it tends to shed at high pH. So S1 shedding is one thing. What happens? So I've thought about that too. When it goes in through the endosome or through the cell, that S2 cleavage, I don't know what happens to that piece. And it's really hard to find that piece. I see in a couple of papers from Gary Whitaker, he can actually find it on a gel, but mostly you don't see it very much. Because it must be happening just, in the endosome or just at the moment of entry.
Right. And with the S1 shedding, the actual S1 shedding, is there anything known about where that S1 goes?
In the medium. I don't know. In the extracellular space. I don't think anyone really knows, but it does its job by attaching to receptor. And by the way, for MHV, the receptor binding domain is at the amino terminus, not at the C-terminal part of S1. I don't know if that's meaningful for anything.
What is the receptor for MHV? Is it ACE2?
Oh, no. CCAM1, carcinoembryonic antigen. It's not a protease, like many of them are proteases, but so that's another whole really interesting thing, right? Because MERS uses a completely different receptor from SARS, causes a similar disease. NL63 uses ACE2, causes a very mild disease. Receptor
229E uses something completely different.
Aminopeptidase.
Yeah.
And OC43 uses carbohydrate, doesn't really have a known protein.
But I thought that there was a preprint that talked about the S1 cleavage product being a decoy or something that Vincent was sent. But
maybe, I don't know.
Yeah, but I think it's actually interesting, the idea that they found the furin site mutated in some isolates from patients where it's not cleaved anymore. Where it wouldn't be cleaved
from
anymore.
COVID patients?
Yeah, from SARS-CoV-2 infected patients. So the question then would be, what is the relevance of that?
Well, I read I don't know.
Yeah. So you can probably still fuse and bind, right? But if you took MHV as the model, you can still fuse, or enter and infect.
You could enter an endosome.
Yeah. Like Vincent's paper in Nature's, he engineered it, but it's actually that patients' isolates have no furin site. So maybe they don't spread down the track by cell-to-cell fusion or something.
Yeah, and also there's a paper I can advertise for one of my former trainees who's back in China, Rong Zhang. He also did a paper where he mutated the SARS-2 cleavage site, and he shows that it doesn't spread in hamsters.
Yeah, I think I sent both you when Vincent invited you and he came down the hall and he said that you were coming on. I think I sent you both that Nature Communications paper, because then we talked about how RSV, there are some clinical isolates of RSV that don't use furin, but they use this calcium-dependent peptidases that are in the ER as it traffics from, or it goes through the maturation of going from the ER to the Golgi.
Oh.
So that particular virus might cleave by another.
Yeah, that's what I'm thinking.
Do they look at that? Someone look at that virus in cells?
No, but I'm wondering how much, if you get rid of, so you might not see it easily like in the particle, but you might see it as the particle is assembled and matured if you took like different cell fractionations, like if you just looked at the Golgi, if you just looked at the ER, blah, blah, blah.
Or just if you looked at lysates.
Yeah. But I don't think anybody does those kind of experiments anymore. Unfortunately.
I'm going to do them. I want to look at what I want to, because, the paradigm is that for SARS-2, that TMPRSS2 is required at the surface for entry in lung cells anyway, in lung cells. But we don't know that's true in all other cell types. In fact, there's a really interesting paper I was just looking to where for MERS, where Tom Gallagher mutated the cleavage site, and he shows that those mutants replicate quite well in like human airway epithelial, but not in Calu-3 cells. So again, it's really cell type dependent.
Yeah.
So Susan, when you prefusion stabilize spike for these vaccines, right? So that is going to prevent S1 from falling off, right? It's not going to be cleaved.
Yeah, I think so.
So I presume viruses with that would not be infectious then, right?
I think not. I was actually wondering about that as well.
Okay. So for SARS-CoV-2 then, both sites probably have to be cleaved. And if you change the furin site, as Amy had said, Vinit's work, it still can infect, although poorly. So something else must be cleaving it, right?
Yes, I believe that. And if you look at Gary Whitaker's kind of review, it'll tell you that, what is a furin site anyway? It's a bunch of basic amino acids.
Yeah.
And trypsin, it's not like a specific sequence. And if you look at them, I was looking at them the other day, the ones for MERS and SARS, and actually Gary Whitaker actually rates them with numbers as how good they are. They're not that great compared to like, HKU1 and some of the bat viruses. So
5-star site would cleave.
Yeah. And just again, back to MHV, the JHM, the one that's super cleaved, has an RRHRR, really basic.
Which I think you first reported as the leucine zipper, right?
No, the leucine zipper was the heptad, the furin domain, I think. No, the fusion domain.
Oh, okay. I got it wrong.
I don't know that we had the right place. That was a long time ago.
Yeah.
So the paradigm right before was that the fusion peptide is revealed by that first cleavage, it would be like you cleave with furin and there's the fusion peptide. But it turns out for coronaviruses, the fusion peptide is more downstream and more internal. So you have to have that second cleavage.
Yeah. Because actually Vinit's paper, there is not a huge attenuation. I think it's a log lost.
In the lost furin site?
When he gets rid of the furin site, I think it's only like a log, maybe 2. So it's not a huge, the disease is somewhat attenuated in his animal model, but it's not like it went from, 100 to, 3.
So it can go either through the endosome or be cleaved by a different protease.
Yeah.
Either way, I think is possible.
So what our listeners should take away is that this idea of entering either at the plasma membrane or in an endosome, which was very much publicized recently for this virus, is nothing new for coronaviruses, right? We've seen it before. Susan,
old, yeah.
What about inhibiting these proteases as an antiviral approach? Is that being looked into? Do you know?
Well, I think that it's kind of flawed because of it's. In some ways, if you inhibit TMPRSS, you kind of force it in by the other route. So I think you'd have to really inhibit both routes. So I think that's why hydrochloroquine didn't work, for example.
Yeah.
And also different cell types are going to be different. Like one of the papers suggested that SARS-2 was really clever because it could get in through the TMPRSS pathway. How does it work? it could go through either pathway, say in the airway, but maybe other organs or other cell types would require it to have the cleavage. But again, we're looking for the virus, and we don't really know. Like one thing that Amy probably remembers is one thing that was sort of interesting is we did a lot of stuff in animal models. So we looked in the liver and the brain lysates at spike proteins from MHV, and they weren't cleaved. So we concluded from that those two organs didn't, and that would make sense in terms of how our persistently infected glial cell mutants didn't cleave, because maybe they don't need to cleave. It's not, cleavage isn't important in glial cells.
Mm-hmm.
Yeah, you have I mean, so I'm interested because I'm interested in neurotropic viruses, and EV68 goes from one site to the next site in various different ways, and we're not clear how it works. But there's like no cleavage for like primary glial cells or astrocytes and hepatocytes for MHV and fusion.
Except for MHV3,
yeah,
it was a very hepatotropic strain which seemed to cleave somewhat in the liver. So none of this is absolute, really. It's all relative.
So is cleavage a good substitute for seeing if the virus is reproducing in a particular organ or not? No,
I don't think so.
Okay.
Because it depends. I think, like, for example, here's another experiment. We took the MHV2 cleavage site, the non-cleavage of MHV2, put it in 859, which is a cleaved strain. That virus didn't cleave, but it also was non-infectious right? But if we selected a non-cleaving mutant, it was okay. So again, yeah.
So Susan, where besides?
Wait, 1 second.
Yeah.
1 second. So her example now, where you take a cleavage site and you move it from one virus that is infectious and you make it into a new virus that is non-infectious. That I know that Vincent has been answering a lot of questions, and I'm sure you have too, about the idea that it's lab strain, derived. The virus is lab strain derived. But this just goes to show you that we really can't trick Mother Nature. We thought we had it all worked out, and now we did something and it didn't work like we anticipated. So the probability of this idea that it came from a lab is just even more supported by the research that you can't just easily do these experiments and have it work as you thought it would.
Let me tell you, this is what I wrote to Vincent, but I think it's really relevant. And all this work is published in the Journal of Neurovirology, which is a very low impact journal because nobody else wanted it, because who cared about it?
I've read a ton of it. I'm glad to know, like, I'm the esoteric reader.
59 and JHM, two related MHV strains. We took S1 of one and S2 of the other, and we put them together. So these are putting together subunits that wouldn't be covalently linked anyway. And we made a virus. We made a bunch of viruses. We swapped all kinds of things in the spike for these two strains. And all the viruses were viable and they replicated quite well, but they were all dead in animals. And so, we tried as much as we could. We wanted to map what was so neurotropic about or so virulent about JHM. And we really couldn't. And so that's why I think that one of the reasons, on top of all the other ones, why I think that it was really not plausible that someone designed this virus. I mean, it just doesn't make sense.
Yeah.
So I'm interested, these recombinants, if you like, or engineered recombinants, they were okay in cell culture, but they lost their pathogenicity in animals. Do you have any explanation for that?
No, it was very puzzling because they just seemed quite robust in replication. Some of them we moved the hypervariable domain, what confers really high neurotropism to the other virus, and I think maybe it became a little more neurotropic. But it wasn't easy to take even what seemed like functional domains and swap them and get it.
This speaks to the whole issue that I think the lay public doesn't understand, which is that there's so much we don't know. And so trying to make predictions about how things will behave in any organism, much less a couple of different organisms is, it's out of the realm of possibility at this point. We don't know enough about what causes disease in humans or what causes transmission in humans to be able to engineer a virus to behave the way we would want it or not.
But in the movies, the scientist designs something and it works exactly in plot logic. I mean, everything fits together.
Yeah, but in 1957, Maxwell Smart spoke into his tie and to his belt, literally. So today, my technician and we were discussing how I have a shoe fetish, and I wanted a shoe phone when I was a kid, and she didn't know where it came from. And it came from the fact that I used to watch these reruns of Maxwell Smart and Agent 99 and stuff. And the one we looked at today, he's talking into his phone and he says, hold on, Central, hold on. I have to get my handkerchief. I have to get my eyeglasses. I have to get my belt. Right. Oh, my garter's falling down and it's rigging. I have to get all these things. And you go and you look at it and it was digitalized and colored, but it's from 1957.
So, Alan, are you saying people are influenced by science fiction movies too much?
Yeah. I mean, these are the stories we tell ourselves as a culture, right? Mm-hmm. And the stories that we tell ourselves as a culture about scientists is that they have enough knowledge to manipulate things in the world. And it's true. I mean, we've got genetically modified crops. We've got, these tremendous technologies that have been developed and that work. But what doesn't fit into the story is the limits of that understanding. And this is what we're talking about right now, that, we can genetically modify viruses in the lab, and that's done all the time. But there are constraints on it, and those constraints don't fit in with the story. And so the problem is that you get the story in conflict with what's possible in reality. And then that spins off these conspiracy theories of people who got the story but didn't get the complexity of the reality of it. And because it's a better story, it carries.
Well, that's why I'm still waiting for my shoe phone. That goes along with exactly what we were talking about and previously about the fact that, people have this idea that science is just a set of facts in a textbook, and it's sort of like received wisdom. It's not something that someone actually had to do an experiment and figure out. And that, it's not something that we know everything that's in the textbook and someone just gives it to us.
With all we know about SARS-2 and all the hundreds and all the new coronavirologists, we still have no clue why it spreads asymptomatically or presymptomatically. Right. We really don't. We have no idea why it's different from SARS-1, which I think is probably one of the biggest, most important questions to answer.
For sure. But Susan, the dialogue, the dialogue has slowly shifted from it was made in a lab, which I think people are beginning to realize that probably it didn't happen. Now it escaped. They were doing experiments on something and it got out by accident. So how do we deal with that?
Well, I think that Peter Daszak's going to have a report soon from his China. Seriously, I heard he talked a little bit about it in his call to NIH. That'll be they really looked at that. I think the only way to it's hard to disprove something didn't happen, but I don't think there's any evidence that viruses like that were present in the lab in Wuhan.
Yeah,
I think anyone in the lab in Wuhan was reportedly to be sick. I don't know, but yeah.
Did you know Zhongli Ji, right?
I met her. I went to a meeting at her, invited by her once. Yeah. And I think I respect her a lot. And I think most people do.
I say, so if she says we didn't have these viruses in the lab, you believe it, right?
I believe it. Yeah. There's always a possibility that there was something that they weren't aware of in some bat carcass they had or bat poop or something like that. I don't think you know. But
I would also just point to the common sense notion that I think it was Ben Franklin who said 3 can keep a secret if 2 are dead.
Yeah,
and so here we are with another conspiracy theory that would involve, at a minimum, a few people in the lab would have to have known that this virus was being worked on. And that would now be the most inflammatory secret on the planet, possibly in history. The notion that even with central control in China and everything, that could be used to suppress that, the notion that could be kept secret is absurd to me.
And I think that Dr. Xi's lab operates like the labs we're used to.
Sure.
Yeah.
Yeah, I mean, and scientists, I mean, we're horrible at secrets and we're horrible at agreeing on things. And you're not going to have a conspiracy of people who worked in a basic research lab.
And the other point about recombinants and making viruses is that when you make a virus, you really do have to start with a backbone virus. So there would have had to be a similar virus somewhere.
Yeah.
From the thing to have come from. It doesn't. You just don't de novo figure out how to put together 30,000 nucleotides.
Yeah.
We can barely put together 7.3.
But the fact is that this is probably not going to go away completely until a highly probable spillover route is identified. Okay, which I think is closer to the case with SARS-1, for example, because you have viruses in bats that are even more closely related to SARS-1, and plus you have the intermediate host and et cetera. That's a much clearer track, and maybe something will come out of this WHO visit to China that will lend more credibility or reinforce or describe in more detail a possible path for the spillover event.
We have two examples with SARS and MERS. Both of them have ancestral closely related bat viruses, and both have a really well-identified intermediate species. And in the intermediate species, the virus is much closer to the human virus. So. Right. And, it took time. They didn't figure that out right away. Right. So I wouldn't be surprised if Peter Daszak and his group figure it out. But.
And that's a great point, that we've got these examples, these very, very clear examples. And I don't think anybody has promoted a conspiracy theory that MERS came from a lab. I mean, that just, you see it in the camels. You see it. The whole story is worked out there. Or SARS-1. Right. And so we know this is something that happens. And in fact, it's been discussed for years.
And after SARS-1, we knew that there were viruses in bats that could interact with human ACE2 receptor even. It didn't even have to be an intermediate species. So as scary as that sounds, it really does. I'm convinced it's from a bat.
Alan, I really like your rap about the secret. I hadn't thought about that before, but the notion that anybody could keep this a secret, that's even less probable than constructing it from scratch in a lab.
Well, you guys did Limfa's paper where he goes out in the field and he looks at the 4,300 square kilometer from Japan to Southeast Asia. And he finds, 100 viruses that are SARS-CoV-2 like.
Yeah.
And the probability of that just, I made something in a lab and now I find viruses out in the wild that are like that, and it's not that few, is, not plausible. But this is kind of like the idea of, I grew up partially in New Jersey and people are still talking about, where's Jimmy Hoffa buried? It's like the conspiracy theory of New Jersey. It's,
Well, I mean, in that case, we actually know that Jimmy Hoffa was. There's a very high risk plausible mechanism for Jimmy Hoffa met his end.
Susan, but people are still talking about,
of course. Well,
and
I
like
haven't
the
heard it in a
conspiracy
while.
theory of like whether or not the FBI killed JFK. I mean, there are all these crazy things, and we had Lee Harvey Oswald in jail and we identified him and the gun and various other things.
Susan, what do you know about this WHO trip? Do you think they're going to come up with anything? I can't tell from what I'm seeing in the press.
I really don't know, but I know that Peter said there would be a report very soon.
Yeah.
I don't know. He did say that it was a really good interactions with the Chinese. It was good. It was good.
Because as you know, many people feel it was kind of a conflict to have him go in the first place because he gets money to go there and sample, right? So he would
These are always the situations, So, in government appointments and everything else, very often the people who are absolutely the most appropriate are the people who are most likely to have a conflict of interest. So where do you draw the line? How do you do that?
We're better suited to do that in a way.
Yeah, exactly.
I think he's great. Lin Fo Wang is a good choice. Marion Koopmans, I know those three, and I think they're all terrific. You should have gone, Susan.
Yeah, and Dr. Shi.
Dr. Shi, yeah.
Well, and there's also the protective mechanism that if you get three scientists in a room, they're not going to agree on much. So
True.
if you have these several prominent scientists going and doing this investigation, and they come to an agreement on something, it's pretty clear that that's the deal.
Susan, you wrote in the email yesterday something like Hillary in the pizza parlor. What's that about?
You know this rumor about Hillary Clinton having, I don't know, doing something to children.
Oh, yeah. Okay.
That's what I mean, this kind of stuff to me is that ridiculous as it's fantasy. It's paranoid fantasy.
Okay.
I think.
Yeah, I thought it was Hillary Kaprowski. Sorry.
No. He's an interesting character. He was an interesting character as well, but no, I don't mean him.
Okay. So another thing I wanted to ask you, which relates to what we were talking about before, besides the respiratory tract, what other site in people do you think is a site of reproduction of the virus? For sure.
Okay, so I'm not really good at this. I know that in our own lab, we've looked at infection of cardiomyocytes, and they get really well. And other people have seen this too. It replicates virus really well, and it gets lots of cytopathic effect inclusion. So that site, the brain is controversial, right? It looks like there's some evidence for antigens in the brain, but whether the virus is actually there or replicating is still not known. Mm-hmm. It's clearly CNS effects, but maybe it's all inflammation. Maybe it's all from the periphery. That's really unknown.
What about kidney? What about kidney?
I was going to say,
I don't know. I think probably, yeah.
Mm-hmm. How about GI tract?
Certainly people get GI symptoms. I would think, yeah.
But we don't. I've never seen good, in situ reproduction evidence.
People have done like iPSC derived different kinds of cells, and I think it probably reproduces there, but I'm not sure.
Yeah, but iPSCs are. So I have a problem with some of that work. So iPSCs go to a certain level of termination of terminal differentiation. Especially like when you're talking about the brain, they go to a certain level of terminal differentiation because you're basing everything on the intrinsic pathways of those cells to determine your final adult version. And that's not how at least the brain works. Like there's a lot of intrinsic signals throughout the 6 layers that influence and diffuse great distances between the lobes of the brain and other organs like the penile organ in the brain and various other things that influence how they finally do terminally differentiate into the adult. So what you extrapolate from those is not 100% probably representative. So you have to be very careful about what you're saying. I think
there's always a caveat about that. Yeah. The other kind of thing I keep thinking about, this work from about genetic deficiencies and interferon signaling, and that can really make a difference in viral spread, at least for MHV. If you have an immunodeficient mouse in some interferon pathway, the virus will spread to organs it would never infect. So I don't know if that has anything to do with more severe disease in humans. But
well, you see that for polio, right? When you took out when we take out IFNAR, it's all over extraneural tissue. Yeah. But then, you have Perlman's observations about MERS, where if you interferon too late after the first round of replication, it's totally detrimental to the organism. It enhances the severe pathology.
What about the long haulers and RNA in the nose? That's kind of interesting.
Yeah.
How long?
What's that story?
Well, just that long haulers sometimes have, I don't know the details, but have RNA detectable in their noses for a long period of time. I don't know what that means. I don't know if it means there's any virus there. I think that we have to be really careful talking about using RNA as a surrogate for infectious virus.
Yep.
Sometimes hangs around.
Yeah, I think Diane Griffin is a really big proponent of the idea that RNA is very persistent without any virus replication, especially in the CNS, like from her measles and Simbus work. Like that's a ton of what she does is, this pathology was, 7 months after the virus replication we don't see any virus replication. But here we have this RNA, and this RNA is, I don't know, sensed or not sensed.
MHV does that. Yeah. The lifetime of the mouse, it stays in the brain, but not in the liver. So there is something more brain-like, brain-specific about persistent viruses in general, LCMV, measles, MHV. But the mechanism by which RNA hangs out without virus is really mysterious as well.
Yeah, I agree that is absolutely fascinating. How exactly RNA is persisting in all of these models. So you also were mentioning different types of cells where SARS-CoV-2 or coronaviruses have been thought to replicate. How do you feel about the data about replication in leukocytes, say the macrophages or T cells or any of those?
From the little bit we've done, we couldn't get replication in macrophages. I don't know about T cells. I don't know. Is there ACE2 on those cells? I don't even know.
I think there is. I just know I've seen some reports that that started out sounding okay, and then as you got into the details, were less okay.
I think,
The abstract giveth, then the data taketh away.
Yeah, basically.
I think that's true for a lot of stuff now. You have to really read it carefully. So.
Speaking of persistence of virus, the Ebola outbreak in Guinea that we talked about, that seems to have originated from someone from the last known cases, 2016, and stuck around for 5 years.
That seems so crazy.
There are 12 base changes. There should be over 100 a year, I think. And so what is this? The virus just sitting somewhere and not degrading and not replicating? It's just incredible, right?
Well, at least for MHV, we tried so hard to get virus out of those brains. We even mashed up the brains and injected into another mouse brain, which is a super sensitive assay. Not one PFU came out. So it's really
Is that like SSPE and measles? Do you have nucleocapsids only? And
I don't know, not much protein, but we definitely saw plus and minus strand RNA and messenger RNA. So it wasn't just completely static. I think it was making RNA. But I don't know that for sure.
You were going to say something?
Did you find polymerase? Otherwise, how else would it make RNA?
You mean enzyme itself? We didn't.
Yeah, would you find RDP? Because otherwise, how is it making?
I could never detect protein. It's too low level.
But
you detect RNA by PCR, but that's about.
No, but could you make an extract, throw in some nucleotides, and have it program to synthesize a small fragment?
I highly doubt it. If we couldn't even get one PFU out of the brain.
Yet by PCR, it's there for the life of the mouse, right?
Yeah, pretty much. Yeah.
Wow.
It's in quasi-species, as everybody talks about. It is. Like there's old papers from Connie Bergman and Susan Baker just looking at sequences, very old papers.
Well, it sounds to me like there's a lot about viruses we don't understand, isn't there?
Yeah, people ought to work on that.
I wanted to ask you one more thing. This is a paper you published after your last visit, which I think is in press now, you said.
I accepted yesterday, like Wednesday.
Very good. SARS-CoV-2 induces double-stranded RNA-mediated innate immune responses in respiratory epithelial-derived cells and cardiomyocytes. Give us the rundown on that, please.
Okay, that I'm really excited to talk about. So basically, what we've shown before from MERS and MHV is that they really, really shut down the three pathways that we focus on: interferon signaling, RNA cell, OAS RNA cell, and PKR. Those are all double-strand RNA sense pathways that are induced during infection. But so if you look in MERS, for example, you see almost no induction of any of those pathways. And the same for MHV. And when we looked in SARS, we were surprised to see that both RNA cell and PKR were both activated in. We looked in various cell types. I mean, most of the work we did in the cell lines, but we looked in iPSC-derived AT2 cells and cardiomyocytes. But we also, one of the systems I really like that we're going to do more is nasal cells. So these are cells that our ENT surgeon collaborator, Noam Cohen, has a huge collection of from patients. And so that's the first cell that's infected. So I think it's a pretty important cell. So anyway, we see very little interferon induced, but we do see these other two pathways activated. And I think that's pretty interesting. And it turns out that SARS doesn't have, like MERS has a double-strand RNA binding protein, kind of like an S1. And it also has, and both SARS and MHV have my favorite antagonist, the phosphodiesterase that we first talked about in MHV. So SARS has a homologous one. I mean, MERS, sorry. SARS doesn't have them. So MERS has two very potent antagonists that are absent from SARS. And SARS has other antagonists that I don't know so much about yet, like ORF6 and ORF3A have been reported to be interferon antagonists. But functionally, we don't see... We compared them directly to MERS, and what we found was that SARS-2 behaves more like a MERS mutant lacking its antagonist. So I don't know what it means, but it's pretty different. So that's essentially what that paper. Oh, and then the other thing, important point I think that we've shown with other viruses is that so a lot of times RNA cell and PKR pathways are viewed as downstream of interferon. You induce it. It upregulates OAS, it upregulates PKR, but really they're independent pathways. So if we take a MAVS knockout cell, which makes very little interferon, we can still activate these two pathways. And that's what SARS does. So that's basically what that paper says.
Yeah, I was going to
Is there any way you can assay those pathways in an animal model of infection? Are there...
Yeah, I mean, we looked at, we did a lot of we had a knockout mouse for RNA cell and for PKR. So you can look at them that way. You can say that if you knock out one of these pathways, does the virus, like, for example, MHV, this is my favorite virus. If you mutate the phosphodiesterase, that's the enzyme that completely antagonizes the RNA cell pathway. That virus is completely dead in wild type mice. It causes no liver disease at all, but interestingly still replicates in the brain. But if you take an RNA cell knockout mouse, that mutant completely recovers. So that's one way we looked at it. And the same thing with PKR. There are PKR knockout mice, and there are double PKR OAS RNA cell knockout mice as well. So then there's this enzyme called EndoU. This is a really interesting enzyme for all coronaviruses encoded. It's NSP15. And it cleaves single-stranded RNA. We think genome RNA. Susan Baker has a different result, but its downstream result is that you get less double-stranded RNA accumulating. So our data suggests that it actually sacrifices its own genome at some point so as to produce less double-stranded RNA. If you knock out EndoU in MERS, in MHV, you get activation of RNA cell and PKR. You have to, and if you knock it out so we have another paper we're working on now. So what we found out is these in MERS, we have NS4A, the double-strand RNA binding protein. We have a phosphodiesterase, and we have an EndoU. And if you knock out one at a time, not much happens. But if you knock them all out or two together, then you get pathways. So MERS in particular is really set up to shut everything down. So maybe that's why it's more virulent. I don't know.
So when you are separating out the effect of interferon turning on RNA cell and PKR versus sort of the virus directly doing that, have you looked at whether that's due to, is that MDA5 dependent or RIG-I dependent?
Well, coronaviruses, at least we found many, many years ago, only depend on MDA5. Adolfo recently had a paper too suggesting that for SARS too. So I think it's pretty much MDA5, although there's one paper that suggests RIG-I. But basically, if you knock out MAVs, you get really little interferon produced, and the pathways are still active. And what we found in the mouse system was that it's the sort of basal level of expression of OAS or PKR that matters. So if you treat cells with interferon to upregulate any of this stuff, you turn on all these other antiviral factors, and you don't. It doesn't matter about RNA cell or PKR at that point. It's a really good thing for initial infection. Like as soon as the virus gets in, before it has a chance to induce interferon, the double-strand RNA is going to turn on these other two pathways. And of course, it's only in infected cells that they're going to be turned on, whereas interferon is going to turn on antiviral activities in other cells. So it's really good for that, for early infection. And also I think for viruses, so many viruses shut down interferon and don't manage to shut down these other pathways. Like Synbis induces tons of RNA cell activation and PKR and still replicates well. So I don't get that, but it does.
But PKR also leads, it's not traditionally thought of as, or at least in my mind, it's not thought of regulating type 1 interferon. It's really gamma.
No, it regulates transcription.
Right, but
it regulates, it significantly regulates the translation of interferon gamma. Yeah, what's the guy in Israel who, the Ray, what's his name, with the hair, with the stem loop and interferon gamma PKR? Vincent, you know what I'm talking about.
Yeah, I know.
But PKR does a lot. It knocks down.
Yeah, but interferon gamma, but so you should actually see some kind of phenotype in T cell recruitment or something or other downstream in an animal, I would think.
Yeah.
Because interferon gamma should be either deficient or you should be recruiting or somewhat, it's linked somewhere to T cells. My mind is blanked. But
yeah, no, all of that I agree. I was thinking more about the fact that type 1 interferon could turn on PKR. And so I was on that end instead of the after PKR.
Yeah, I'm on the after end and then the pathogenesis in the animal. And so whether or not you would,
But PKR is really interesting. We're really fascinated by it because it turns on PEI. It phosphorylates EIF2 alpha.
Yeah.
And it happens in SARS, but not in MERS. Yeah. So one of these viruses, and that knocks down protein synthesis. In addition to NSP1, that SARS also knocks down protein.
Yeah. I mean, so my friend identified, so Mauro has spent his entire career looking at EIF2 alpha phosphorylation in the context of learning and memory. And one of his findings is, it's a Cell paper, I don't know, maybe 10 years ago, where PKR is regulated by EIF2 alpha or regulating EIF2 alpha. And the downstream effect of having too much or too little interferon gamma in the brain and learning and memory and savant syndrome. The details are a little hazy because I haven't thought about this stuff since we left Sonnenberg's, but that's it's really been, and it has some link to ADAR and some other interferonopathies or something like that.
That's the other part of my lab.
Yeah. So it's one of those integration points that is very confusing and hard to.
But PI2 alpha is also phosphorylated by PERK, and so been looking at that recently. And again, that happens with SARS and not with MERS. So I think PIF2 alpha is going to be really important.
Yeah. So you can also look at GCN4 and HRI, the 4 kinases that phosphorylate EIF2 alpha.
So Susan, what is it? EndoU is the name of this is an endonuclease, right? And
yes.
This degrades single-stranded RNA to prevent activation of double-stranded RNA sensors, right?
Yeah. It's kind of in a way.
And now, if you take out EndoU from the genome, can the virus still reproduce?
The virus, if you put it like in a Viro cell or something like that, where there's not much double-strand pathways, it's perfectly infection. Yeah, it replicates less well, say, in a macrophage, where there's pathways getting activated.
But Viros are lacking the chromosome 8 portion that encodes all of the interferons.
Right.
That was my point, that if you don't have a response.
But in a cell that has an interferon pathway, that virus would be dead, right?
Not dead, but it would be weakened.
Weakened.
Evaluated.
Is this a potential drug target at all? Is it
I think so, along with, you don't get me started on that. There are so many drug targets in coronaviruses. Yeah. I mean, Mark Dennison's paper that you've referred to before, the recombination one, SP14, that's a really good drug target.
Yeah.
And you were part of a screen of FDA-approved compounds for this, right? Did anything come of that?
There was a very little part of that screen. So I don't know anything more that's in the paper. We were really just collaborators that's Matt Freeman's work. We applied him with the 8549 cells. The reviewers wanted human cells to be used.
So as to the best of your knowledge, I sound like a lawyer. Are there any? So we heard there's this Merck drug, molnupiravir, right, which is in phase 2. Is there anything else in more advanced trials? Any other antivirals, say?
I really don't know.
Yeah.
But there's so many targets. There should be.
Yeah. There are lots of targets. Yeah.
A and 1B. There's like, it seems to me like, the combination of a protease inhibitor and an RDRP inhibitors work for HCV and HIV, right? That seems one strategy. But then we have these other, this exonuclease, this endonuclease. There are 3 capping. Well, I guess there are 3 capping enzymes.
I think from what I've seen, a lot of the focus for industry has just been so much on vaccines that other than Merck, there just hasn't been a whole lot of movement on the antivirals, because the perception has been, well, the vaccine is the way out of this pandemic. But longer term, I think antivirals could have tremendous potential. And I'm actually really excited about the Merck one. They dropped their vaccine program, but the antiviral program potentially is something that could be given, in maybe even prophylactically or that kind of thing.
I thought Nevin and Adolfo were doing a lot of were screening at least FDA approved drugs already, and very few have fallen out of that.
You're not the only ones. I mean,
no, it's just that their names come, I've known Adolfo for years, and we're sitting in New York. So,
the strength
and Adolfo's everywhere. So,
the strength of doing the FDA approved drug screen is, of course, if you get a hit, jackpot, it's already approved. You just go and tell people to prescribe it. And often the whole profile of the thing is well known. The drawback of that is that there are very few successful antivirals that are in that group of FDA approved drugs.
Yeah, that's what they found.
What Susan is talking about with all the targets in coronaviruses. I mean, this is a target rich area, but they're targets that aren't likely to be hit by anything that's already been approved, because this just hasn't been worked on. So it's
a long-term thing, as you said. I totally agree. I mean, if we're going to get another coronavirus in 10 years, we have time to develop it. I mean, I think some drug companies are working on it, actually. There's definitely corporate activity on this. It's just, it's not receiving the kinds of money and emphasis that the vaccine work has received. But I'm hoping that one of the things to come out of this pandemic is that we'll see serious effort on targeting all these types of targets that you're talking about, because as you say, the next pandemic could very well be another coronavirus, and wouldn't we want to have some drugs for that?
Well, maybe Vincent should have Ralph back with his collaborators and colleagues who are running Ready.
Yeah,
because that's the whole purpose of Ready. But to go from a novel, chemical on your shelf to develop an antiviral, it's not that exciting. Yeah.
Well, Alan, are you saying
a lot of failure somewhere along the line?
So Alan, if we had put $24 billion into antivirals, we would have some, right?
Sure.
Can I tell you a story too? And not back in 2000, and after SARS, I forget what year, I sold a green GFP expressing MHV to 2 drug companies because they were so hot to use MHV to screen, and then it all went away. I think they closed their antiviral department or whatever. So we really should remember that stuff, that had they continued on that stuff.
Well, this discussion goes back to, Vincent had repeated rants in the beginning of the pandemic about how we should have been prepared for this by years worth of antiviral drug screening. And in fact, this just reemphasizes that there ought to be an effort that's like CEPI. CEPI,
well, that's what Ready is.
Yeah. Ready is, if I'm not mistaken, Ready is
host.
Sort of focused on host.
Yeah.
But it ought to be but nevertheless, same sort of thing.
Yeah.
Antivirals in general.
But it's not just the pharmaceutical companies. I'm sure that if you talk to Mark Dennis, and I'm sure if we talk to even Susan Longer, they will tell you that they wrote hundreds of grants to do this at the NIH, and the NIH said, not discussed.
Well, that's the point. Is that there ought to be
those grants.
the money.
Yeah. I mean, I would
to do this.
If I were a study section reviewer and it turned out I was the one, I wrote that review, I'd feel so guilty. I'm not sure that I could. Yeah.
So
might have to leave.
When we talked to Mark Dennison last year, that's what he said. He said, I can show you dozens of grant applications that never got funded for making antivirals. Yeah.
Kept persisting pretty much during that time.
Yeah, maybe. But so Susan, do you think, the question is, do we learn a lesson or is it going to go back to the same old?
I hope so. You don't think so? I don't know. I don't know. I hope so. I mean, NIH should be part of this story, right?
It has to be.
Yeah, for sure.
The thing is that it has to be an effort that is where the funding mechanism is different.
Yeah, I think.
Than the standard funding mechanism. Something, that comes out of an agency like BARDA or something like that, that's going to be able to fund stuff that study sections would find boring, or that pharmaceutical companies would not want to invest in. To take them up, do all the appropriate screening to get stuff just to the point where when something comes along, we have a shelf full of candidates to go to.
I think we have a window of optimistically maybe about 5 years where these types of things can be set up and we can maybe get to the point where we've got some antivirals in the locker that have been through some phase 1 studies and maybe even some phase 2 studies before we go to our usual cycle with this type of thing where we say, that pandemic was a one-time thing. We can use this money to build more bombs or something, and the money goes away. But if we get that going within the next few years and we get that amount of progress, then 15 years from now, when the next pandemic does come along and it happens to be a coronavirus, or hopefully this will be a broader effort looking at the most likely viruses to spill over, we can say, oh gosh, we cut all the funding for that. But we've still got these compounds sitting around that we use, so we could at least pull those out.
Yeah. I hope.
I think 5 years is way too optimistic.
I am being optimistic. The public attention span is usually 6 months, but because this pandemic has gone on for over a year, I think we may have a little more leverage there.
It hasn't done yet. Right? We're still living with it.
Yeah.
Susan, what do you think is the end game here? What are we looking at?
I guess I'm on the optimistic side about the variants, if that's what we're talking about. I think that they're going to keep changing until everyone's vaccinated. Not until everyone, but until enough people are vaccinated. And I also think it's going to reach an equilibrium. I think it's adapting to humans. Mm-hmm. At some point, I think it'll be adapted unless we start getting people infected and getting statements. So far, I think most of the mutations are just adaptation.
So what do you think in that context then, like about Crotty and Study's recent paper that talks about the fact that none of these variants are in T cell epitopes and the role of T cells and its importance in immunity for coronaviruses would suggest that, okay, so these change, big deal. We're still, doing well.
That's part of my optimism that the vaccines, but then you hear the story about in Brazil where people are getting reinfected with variants, people that have already had the original virus. So that worries me a little bit.
I don't know. It's hard to parse that coming out of there from the press. I'd like to see the papers, and we don't have those. So I'm not, go ahead.
No, I was going to say, I don't think particular variants are like with this thing about closing the English channel at the beginning. Like, it's not the idea of keeping variants out because they're popping up everywhere with similar mutations. Yeah. And they're doing that is because they're giving the virus some kind of advantage. Yeah. So, sense to me. No, so I think I'm more optimistic that as people get vaccinated, the variants will become less of an issue.
Right.
Yeah, the variants are providing a convenient excuse for politicians who've lifted restrictions and are now seeing, like in Europe, the new spike of infections. And, those wascally viruses, it's the variants. It's not because we reopened all the restaurants. Hopefully we will manage to get ahead of it with the vaccine in time.
I've been sort of meetings in Europe, and most of my colleagues in Europe are not vaccinated the way we are.
Wow.
Yeah. And they're relatively young, and they're not particularly getting vaccinated quickly, which sort of surprised me.
Well, I have friends in Canada who will tell you the same thing. I have a friend who's, I think father is 79 or 81, somewhere around there. And my relatives who are of that age were vaccinated like in January. His parents, I think, are still waiting online.
But I'm talking about people that work on SARS.
Yeah. No, I know what you're talking about. But
I think, Amy, didn't we give Canada some of our AstraZeneca stocks now?
Apparently.
Yeah. Well, Susan, I'm going to let you go. We didn't hit everything, but that's fine. We appreciate your coming here. University of Pennsylvania, thanks so much.
It's fun as usual.
And
thanks, Susan.
See you next
Great
time.
having you.
This is awesome.
Bye-bye.
Bye.
Let's do some email. I think, Rich, you should take that first one.
Thank you. Eric writes, "Hi, Twivers. Me again. Just an astronomer who's been listening to TWiV since last February. How time flies. During TWiV 731, you folks discussed the orbits of the James Webb Space Telescope and the Hubble Space Telescope. This week in space, right? Vincent asked where the James Webb Space Telescope was heading, and Brianne said correctly that it was going beyond Earth orbit. It will be orbiting, God, this just blows my mind. It will be orbiting the sun 1 million miles from the Earth at the second Lagrange point. That's a point where the opposing gravities are balanced out. So it sits there. This is not similar to the Hubble Space Telescope, which is in low Earth orbit. So astronauts could service it from the space shuttle. They did this 5 times to install new instruments and replace broken equipment. Here's an animation of JWST's planned orbit and gives a link to that. Here's a NASA page that discusses the 5 Hubble servicing missions, 1, 2, 3A, 3B, and 4. All the best, Eric. And I haven't looked at those videos, but I will, okay, because they sound great. Yeah, trucking out to L2
not happening
is not happening soon anyway. Okay.
So they have to get it right the first time, basically.
Yeah.
Yeah.
Okay.
I hope
right, because if you get it a little too close to the sun, then the sun's gravity takes over, right?
Yeah.
Yeah. You can't set it out with a bad mirror.
Right.
Okay. And then send up somebody to fix the mirror.
I guess they've learned, unlike, the pandemic people, right? Or maybe not. Thanks, Eric.
Yeah, thanks.
All right.
That's great.
Brianne, can you take the next one?
Sure. Sanjita writes, "Good morning. I'd like to thank the whole TWiV team and Dr. Daniel Griffin for inspiring me to run a COVID vaccination clinic from my office. I'm a pediatrician in Texas, and after many weeks of requesting, we finally received 100 doses of the Janssen vaccine. We vaccinated our high-risk 18-plus patients, their parents with medical conditions, and educators in our community. This past week has been the most gratifying experience for my staff and me. I wouldn't have dreamt of taking this initiative were it not for Dr. Griffin's pearls of wisdom. Never miss an opportunity to vaccinate and never waste a dose of vaccine. It's a lot of hard work to organize a vaccine clinic from a small community office. We were all exhausted by the end of the week, but well worth it. My nurses were able to get 6 doses from most vials of the Janssen vaccine, so we ended up giving 116 doses. Again, love your show. You have inspired a lot of us during these tough times. Keep up the great work." And this is from Sanjita from Lake Louisville Pediatrics.
Nice story. That's great.
Nice story.
Love it. You know where that is in Texas, Rich?
Yeah, I just looked it up. It's just north of Dallas.
Okay.
Looks nice, actually. It looks like a big old lake.
Yep, good stories. Alan, can you take the next one?
Sure. Steve writes, "Good afternoon, Vincent, Rich, and everyone at TWiV. Thank you for continuing to provide amazing insight. I have 2 vaccine questions. A student just contacted me who has had her first shot and is to get her second shot tomorrow, but she tested positive for SARS-CoV-2 yesterday and is symptomatic, mildly. She approached me to ask if she should get her shot tomorrow. My feeling is to defer as she's already forming a new immune response, but this is way outside my area. I was wondering if the brain trust of TWiV had a thought or if there was a definitive answer. My first suggestion to that is that she should show up for her second shot and tell the people administering it that she has tested positive for SARS-CoV-2, which at least when I got mine, that's one of the questions they ask you. Have you ever had a positive test for COVID-19? So the answer would be yes, yesterday. And
would they send her away, you think?
They may, but I think she needs to disclose that to them and get the official input on it. I do not feel comfortable giving advice on that.
But I think Daniel has already addressed these issues many times
what did he
during
say?
his clinical update. I think he says that you should defer, that you shouldn't go and get it at all. You should wait until you clear the infection and then wait a month afterwards, that you don't even go.
The reason I suggest going is because there will be a protocol for rescheduling.
Yeah.
And
otherwise
if you don't go,
it.
if you defer, you're going to lose your spot, and who knows when you're going to get the second shot.
Yeah, the only thing I worry about is having somebody who potentially is shedding virus walking around with vaccination.
Yes.
Probably if you have a first shot and an infection, probably when you get your second shot is probably not that important. You probably can wait very long periods of time.
Better plan might be to call the vaccine clinic.
Yeah.
Tell them what's up so you don't show up. Because Rich raises an excellent point. She shouldn't go anywhere. She should be sending out for food and staying at home and isolating from everybody. But yeah, I would still urge informing the vaccine clinic and following whatever the channel is for the delayed dose.
So in general,
I think in general, Daniel has discussed this many times on his clinical updates.
But also, if you're sick with anything, they say not to be vaccinated until
not to be vaccinated when you're sick.
Yeah.
And the second dose, certainly of the Moderna, and I've heard this similar for the Pfizer vaccine, it packs a punch for a lot of people. A lot of people have no problem with it, but if you're already sick, you probably don't want to have that reaction.
Well, I was sick for 4 days after my second dose of Pfizer.
Okay. I was in rough shape the day after, not nearly as rough shape as my wife was or as my sister was after their second doses of Moderna. They got the full ride, the whole, nausea, vomiting type of thing. It's still worth it to be vaccinated, but yeah. All right. So continuing with the email, a sillier question. Oh, good. My wife and I each had our first Moderna at the beginning of March. She asked me a question I could not form an answer to last week. Should we get our second shot in the other arm? That is, are there any localized effects? She still has a tiny bit of redness that we want to spread around or concentrate. I'm guessing it does not matter. But then again, I work on viruses that make the oceans go, not the ones that make you sick. Just keep up the amazing work. And Stevens in the Department of Microbiology at University of Tennessee. We talked about this just last week or the week before.
Yeah.
Which
sometimes.
Yeah, but it has come up, and the consensus seems to be it most likely doesn't matter. Yeah. But the trials were done same arm.
I have a hard time imagining that it really matters.
Yeah.
Well, Daniel again has addressed this on his COVID updates.
What did he say?
He says,
doesn't matter.
He says it doesn't matter. It diffuses around. That, I chose to do the same arm because I'm right-handed and I'm very active in the lab, and I don't want my arm to hurt and not work as well as it needs to work on a day that, I'm dissecting out brains.
We're doing plaque assays.
Exactly. Plaque assays are every day. But yes, but listen to Daniel. Review Daniel.
Steve Wilhelm was on TWiV. Remember, Rich, in Germany in 2019, right?
Yep. Steve's one of our giant virus buddies.
Yeah.
He worries about viruses and algal blooms and stuff like that.
Yep. Amy, can you take the next one?
Sure. Sean writes, "Hi, I'm a regular TWiV listener. Much of your podcast goes way over my head, but I listen anyway. From what I hear, the progress against the virus is proceeding much faster in the US than it is in Canada. I live in a small city in Northwest Ontario that is in its third lockdown. We have had over 1,500 cases since January 1st, 2021. There are currently 400 active cases in a population just over 100,000. It seems obvious to me that a good portion of the population is not taking the virus seriously. It is very difficult to remain positive. I have been an optimistic person all my life, but it is very hard to essentially live in a loop. We are not supposed to go out, to travel, to eat out, to socialize, and I have adhered to it. So every day is pretty much like the day before. It does raise my spirits to hear on your podcast that other areas are doing better now. I just wonder when that progress will make its way up here. Our medical officer of health has said about our area that the virus has taken on a life of its own and assume it's everywhere. So it's not good here. If I had to live my life over, I certainly would pursue microbiology with a focus on virology. But at 72, that's not an option. Just hope I am alive at the end of all of it. No question, just needed to vent. Sean from Thunder Bay, Ontario. So he should not feel so bad because I hear from my friends in Montreal that they have not left their houses and are in lockdown, as he describes, with a curfew of 8 p.m. for months on end by now. And most of their complaints go to the government, who don't seem to understand how to run a pandemic efficiently. And the lack of, they also will address the fact that there is a lack of wanting to maintain physical distancing and wear masks and stuff in some areas. But in their area, they say that they're very adherent.
Well, the good news, Sean, is some vaccine is coming your way. And since you're 72, maybe you will get it. Millions of doses, right?
Yes. Mm-hmm. Millions of doses. But so Ontario is actually one of the provinces that is actually, from what I understand from my friends in Quebec, is doing better than Quebec. Yeah. Quebec is the epitome of the disaster, apparently.
I am sad to hear these kinds of emails. Don't lose faith. Just, it'll be okay.
It'll be okay. It'll come to an end.
Yeah, it may take a little longer, but it's being worked on. And yeah, you've got, so the U.S. government has bought hundreds of millions of extra doses. And once those are produced, they're going to start sending them to both Canada and Mexico, I think.
Yes.
Actually, there's another piece of good news, which is that you live in Thunder Bay. Mm-hmm. That looks like it might be a very pretty place to live.
Where's that?
I've heard of Thunder Bay. It's like a big vacation destination.
Yeah, it's essentially on the north shore of Lake Superior.
Ooh.
It's a little cold at this time of year with a lot of snow.
Yeah, sure.
No, maybe not this time of year.
Yeah, but it's like a 6 weeks window. You have to understand it's like a 6 to 8 week window of summer.
Yeah, but gosh, I'm doing an image search and this looks gorgeous.
Yeah,
it is. It's really pretty, but it's also very fleeting.
I only need a week, it's.
Yeah, but I'm sure she wrote this in the middle of the winter when it's dark.
Yes.
I mean, you really don't understand what 6 to 7 hours of daylight is like. It's kind of depressing.
How about 2 hours of daylight?
And the gag in Buffalo was that we had 2 seasons, winter and the 4th of July.
Oh, yep. I've heard that.
In Massachusetts, we say it's winter and road repair.
Yeah, I was going to say, in Syracuse, it's winter and road construction.
All right. Vanessa writes, hello, TWiV team. I heard on episode 731 that dog walking while listening to TWiV is a common thing. That's actually what got me through the quarantine last year. And I definitely enjoyed my long dog walks during the summer while listening to TWiV and my favorite podcast, TWiVo. I heard Vincent say it was too bad that the dogs aren't getting educated as well. I will say at home, I listen out loud on my computer. And the joke is that our dog Mila loves it too. I have proof. The living room futon is her least favorite place to spend her time. and she tends to avoid being around me while I work from home because she resents every minute I'm home and not paying attention to her. However, she almost always comes over to the living room and takes her naps on the futon whenever I'm listening to TWiV and TWiVo. It's become a running joke that she loves Mommy's science podcasts. So there's at least one dog getting educated as well. Thanks for everything you do to keep us all educated on this topic. Vanessa's in Pittsburgh, where it's 34F, feels like 28. 1C feels like -2.
Must be the voices.
Probably.
I must have voices that are pleasing to dogs. For whatever that's worth.
It's good. Let's do one more round. And Rich, why don't you lump the next two together?
Okay. Scott writes, last episode 731, you received an email about a picture of the wrong Vincent appearing on the vehicle screen playing TWiV. I almost emailed you several months ago about this. See photo below I took from 664. Alan appears to have been right. I believe the actor-director is Vincent Gallo. This is misleading. The mislabeling is a recurrent issue on my Toyota. Newer artists are shown as older ones. Jackson Wang is shown as the Jackson 5. Charlie Puth is shown as Charlie Parker. Clario is shown as Charo. Best Scott in Madison.
So they just picked any random Vincent.
Any old Vincent.
Well, I think Gallo.
Any old Vincent will do.
You know, Gallo has some overlap with Racaniello, right?
Yeah.
Yeah, I suppose. I wonder if Vincent Gallo gets pictures of you, Vincent. At any rate. And the next one is JRJA. Is that actually just initials? Or
I don't know. That's what it was.
JRJA, Georgia, writes, hi, Twivers. In response to Suzanne's email on TWiV 731, Alan had it right. The actor and director Vincent Gallo. He's the stock photo that seems to be showing up in her car. No real idea what's happening, but I'm guessing that the name was the closest to Vincent Racaniello that the automated system found. As my first programming teacher told me way back in the early 80s, the computer is stupid. The rise of AI has done nothing to change this fundamental fact. Georgia.
Alan, you got it. Good job.
Okay.
Nice.
There was an XKCD cartoon about this a little while ago that was it had a drawing of a Wikipedia page about this medieval document called the Missal of Silos, which is it's a religious document. It's located in Spain, and the caption on it was, Spain would like to remind everyone to use strict pattern matching in their missile targeting algorithms.
Brianne, could you take the next one?
Sure. Amanda writes, hello, tremendous TWiV team. As a New Zealander who has lived in the US for over 30 years now, I've watched enviously as my friends in New Zealand go about daily life while we have been locked down here. I thought you might be interested in this article in the Washington Post this week, which offers some insight into one of the reasons the COVID response has been so successful in New Zealand. And she gives us a link. She continues, unlike the rugged individualism that is a key component of the self-image of so many in the US, in New Zealand, every school child is taught about mana katinga, or the power of the collective, key to Maori culture. Others have importance equal to, if not greater than yourself, and your freedom as an individual is only as strong as your place in the community.
Man, I knew it. I'm going to New Zealand. I'm done. I'm done with this place. I'm going to New Zealand, man.
That's a great philosophy, isn't it?
I'll
It
say.
I like it. On another topic, I was relieved to hear about the TWiV listener with the odd photo for her car's podcast image. I thought it was just my car going rogue. I thought you might be amused to see my car's idea of you all. And she gives us a photo. And I'm not sure.
Which one is you, Brianne?
That is not Vincent Gallo.
I don't know. I don't know if this is sort of, it's a pageant or.
I don't think it's
I don't think it's the Spice Girls. It's five women in sort of semi-matching outfits.
Must be a group that matches. Saturday. Isn't Saturday the name of the group? Right. Let's look.
Well, as she concludes, look like you're all now social media influencers or some such. Right. Many thanks for keeping me sane this year, Amanda.
Well, if you search for Saturday, you get dozens of girl groups, not just them. So I don't know. That's a common name, I suppose. That's really funny, I think.
That is.
So the tallest one is Rich, obviously. And
I'm thinking it's the Saturdays.
The Saturdays.
The Saturdays. 1, 2, 3, 4, 5 of them.
And do they dress in rainbow colors?
They do indeed.
Yes.
Yeah,
British girl group. Okay.
So why did that come up? Maybe because it was downloaded on a Saturday or it's weird. That's really weird. I don't have this issue because I don't have this thing. Well, maybe I do. I just don't use it. Right. Anyway, thank you for that little diversion. Alan, can you take the next one?
Sure. Tom writes, I've been listening to the TWiV podcast since last April. I'm a district school administrator for a large school corporation in the Midwest. Among my many responsibilities, I'm responsible for our COVID-19 policy and response, including being the chair of our decision-making task force. My group conducts contact tracing, administers rapid tests on staff, and makes recommendations about the degree to which we have in-person education. This responsibility monopolizes the majority of my time. We're guided by science, including a group of physicians that have treated COVID-19 patients, our local health department, and scientists from our local colleges. I cannot tell you how difficult these decisions are. We have well over 15,000 students and 2,000 employees. The decisions we make will impact their livelihood and possibly their life. School corporations all over the country are saddled with these painstaking decisions. Your shows have been a godsend to me. I listen to every podcast. I don't understand everything, but in large part, you have guided aspects of my decision-making. You have had a profound impact. I cannot thank you enough. In episode 731, The Kids Aren't All Right, you delved into my world. I appreciated the conversation. One thing you must know is that there is a real emotional impact on children, and COVID-19 has magnified an ongoing crisis in student mental health. COVID-19 has been like adding gas to the fire. The lack of socializing has caused huge problems. Suicidal ideation and suicide attempts have increased significantly among our student population across the country. When wrestling with the return to school, weighing the danger of the virus is paramount, but so is the mental health of our kids. This is difficult, to say the least. We would love to test students, but monetary and human resources are not available. Recently, I saw this preprint and would like you to discuss it and any relevance with school. And I haven't seen this yet. Says association between SARS-CoV-2 transmission risk, viral load, and age in Danish households. Okay. P.S. have thought about TWiV, the movie. One word, blockbuster. I was thinking of actors and actresses to play the part. By the way, I've only listened to the podcast and have never watched. Vincent is Christian Slater, Rich, Seth Rogen, Brienne, Jodie Foster, Alan, Tom Hanks. Oh, okay. Yeah. Kathy, Glenn Close. That's good. Dixon, played by Dixon, the one and only that could pull it off. Okay. I was hoping for George Clooney.
I'll take Jodie Foster.
Jodie Foster's good.
I thought I would rather have either Joe Pesci or who was the other?
You have the list in front of you. It's on the list in front of you.
No, I don't have the list anymore. Christopher Walken. The guy with the Brooklyn accent. I don't have.
Broderick is another one I've gotten occasionally.
But Christian Slater's okay. Actually, looks a bit like me. It wouldn't be a blockbuster. Sorry.
Tom Hanks is a little old for me.
He's very good. He's a very good actor.
Oh, he's an excellent actor. Yeah. Yes.
Who do you want to be, Amy?
So I
was left off. I don't need to have a person.
Yeah.
There is no person. I was left off. It's okay.
Let's see if we can summarize this.
So I have not seen this paper. It's a preprint.
Associated between transmission risk, cycle threshold, and age of infected cases. So they have data, administrative register data, comprising the full population and all the tests that were ever done from August through February to estimate household transmission risk. That's what you can do in a country like Denmark, right?
Yeah.
We found that transmission risk was negatively associated linear with the CT values of the tested primary cases. Not surprising there, right? Right.
Yeah, so the CT value data, I think, is no news. And
listen to this. Even for
they
high CT, the risk of transmission was not negligible.
Right.
So for CT of 38, they found a transmission risk of 8%, implying there's no cutoff, no obvious cutoff. So that people have said that because CT can be different on different machines, right?
Right.
So
CT is also different by different primers.
yeah.
They found an age-associated transmission risk, which is, I think, more interesting for the school discussion.
So what was that? Linearly increasing risk with the age of the primary cases for adults and negatively for children less than 20 years. So with increasing CT, less transmission risk for children. Is that what I'm getting from this?
Mm-hmm.
Age had a higher impact than CT value on risk of transmission.
Yeah. I'm not. There were just in today's MMWR, for this week's MMWR, there are a couple of papers on relevant to schools looking at distancing, and this is in U.S. schools, which probably more relevant to Colorado. There's one looking at distance that found 3 feet of distance was not any worse than 6 feet of distance for the school that they looked at. And there was another one looking at school transmission. I mean, basically what all these papers have been pointing to, there have been earlier ones, MMWR and elsewhere. What we're converging on is that the schools can be reopened safely if you've got some degree of distancing. There's now this discussion about 3 feet versus 6 feet, but that's coupled with mask use that's actually enforced. And those seem to be the big things. I think lunch is a big issue. You keep the kids spread out maximally at lunch because obviously they can't be wearing masks, and they may be talking if they're at the tables together. Just same reasons we shouldn't be opening the restaurants. But yeah, it is looking like schools can be safely reopened if you're rigorous about your protocols. And obviously testing would be best. The more you can test, the better. Certainly the limitations on money, which are the big deal. That's an issue. In Massachusetts, we have at least gotten funding from the state now, and a lot of schools are doing pooled testing. And so they're able to catch these asymptomatic cases, and they're finding them.
I think, though, in the COVID bill that just passed, there was $10 billion given to schools for testing.
Yes.
Nice.
So I think.
Yeah. So that the money should be available soon. And if to whatever extent possible, testing should be done. Do whatever amount of pooling you have to with the budget available, because I think that's really a critical component. But all these points that Tom makes about the mental health of kids, it's a huge issue, and that's a big part of the school reopening discussion. And I feel for the folks who have to make these decisions.
Well, for the parents. But hey, your school district, Vincent, the question wasn't about money. The question was about privacy or something or other. You said you came back from one meeting. If I remember correctly, the one meeting that you attended, and you said, waste of time. They say you can't test.
Yeah, the state of New Jersey says you can't test. That's it. It's a law.
Because they don't want to pay for it.
No, there was some privacy, and
well, maybe
I bet they're making up excuses.
Maybe
anybody who's waving HIPAA about this is just full of it.
Maybe this 3 to 6 feet. Daniel talked about this, and he thinks it's kind of bogus because they looked at records where schools were either 3 feet or 6 feet apart, the kids, and they looked at the transmission, but they didn't actually verify that you were 3 or 6 feet apart, right?
Oh.
That's just a regulation, and it could be that nobody paid any attention to any of it. So he doesn't think it's worth even. But he said, on the other hand, if you do 6 feet, then you can't bring everyone into schools, and that's the problem. And there's this whole issue, like our emailer said, of getting kids back, and I totally get that.
And it raises the important point that 6 feet is not some limit the virus recognizes. right? I mean, this is not something that was determined by rigorous science. It's, okay, what's a reasonable distance? Okay, we think the droplet transmission, and they drop off after 3-ish feet. So let's say 6 feet of separation is safe, and 6 feet is easy to estimate. And so that's why 6 feet was chosen. Yeah. That doesn't mean that 5 feet is unsafe or that 3 feet is, appalling. And so that's what the discussion is.
So, Tom, this paper basically says that you can't make a cutoff, at least in Denmark, saying if CT38 and above, you're not transmitting, because they do see some transmission. And also that is lower at lower ages, which we sort of know, right, already, because transmission overall is.
But you're only worried about CT values if you're doing testing anyway, which you should be doing. And if somebody's if somebody clears the threshold for a positive test, then send them home and quarantine them and do the contact tracing.
Yeah, I'm not sure what the relevance of the Denmark CT value is in America when the American CT value is very clear that you don't transmit after 30 or 33.
I agree. It's different everywhere.
So,
Amy, can you take the next one?
Virus rates. Dear TWiV team, I am a big fan since summer 2020. I listen to your show in Spotify every day when doing the chores or going to work by bike. I have learned tons of things thanks to you. I am a Colombian living in the Netherlands. As a good Latin, I speak every day with my family, especially my grandmother. We and my family all want her to get the vaccine, and she cannot wait to get it herself. Seeing her at her almost 90 years of age, being a prey to the pandemic fear and confined to her home has been devastating. Recently, some vaccines were delivered to Colombia, and my grandmother will have a chance to get her shots, 2 probably. So probably it is the Pfizer or Moderna one, in the course of a month. We are worried about corruption or poor handling of the vaccine in Colombia. It is not crazy to imagine some of the shots being replaced by whatever placebo, or the storage temperature not being low enough. There are stories of people that were supposed to get the vaccine, but the syringes did not have anything in it. So we want to make sure she gets the real vaccine. I thought of ways to control for that. Would an antigen test being applied after the 2 vaccine shots provide confirmation that she got the real deal? If so, are there any commercially available that you would recommend? Also, we thought that of perhaps giving her a second dose once they were offered by private companies in Colombia. Would a second COVID vaccine produce any adverse effects? I really greatly appreciate your guidance, and thank you for helping fight the disinformation, which is abundant in all social media. Kind regards, Boris. So, it's very upsetting to learn how countries are so corrupt and stuff. I believe that the temperature sensitivity of both Moderna and Pfizer have been recently addressed so that they are much easier to resolve, or not so dependent on these crazy ideas of -80 freezers and various other things. An antigen test could be applicable since these vaccines are for spike, so she could get an antigen test looking for antibodies just against spike.
Antibodies, though, not antigen.
Yeah,
right?
That was my question.
Yeah, antigen would probably not be good enough. Right?
Yeah, not be good enough. And I don't know anything about the commercial ones. Like, I don't know enough about which ones people.
I think what he's saying is when private companies.
No, he says if there are commercial antigen tests available, which one would you recommend? And I don't know enough about that and what is on the market and who's manufacturing them. And so, I mean,
especially
at this point,
in Colombia, I don't know what's available there.
I have no idea. And then he says, would a second vaccine produce any adverse effects? No, it would just be an immune boost, like getting a measles shot at 2 years old and then coming back at 5 years old. So it's perfectly fine. And it doesn't even have to be the same one that she got the first time. It could be if she got Pfizer now, she could go for Johnson & Johnson, whatever Colombia is doing. I don't know what their policy is. I don't know enough about Central American governments or South American governments.
I don't know if anyone does. I think Daniel has recommended some antibody tests, but I don't know if they're available in Colombia. That's the problem, right?
Yeah, I mean, I don't know if you want to send them, like if you could send them, if they're even that available in the Netherlands, that you could package them up and send them as a care package to people in Colombia. No idea.
Yeah.
And we do want to make clear that whatever test, if they can find a test, the antibody test is what you want, not the antigen test.
Yeah,
you want to test for antibodies, see if she's producing antibodies. But the results of that are not going to be conclusive.
Right.
And I would say the second vaccine is a fine idea. If you have any doubts about the first vaccine,
get another one. Yeah.
Just when you get a chance to get it from a trusted source, get her another round of it, or treat it as if she never got the shot.
What was that T-cell test that Daniel was talking about recently?
Yeah.
Is that not commercially available?
Yeah, I'll pull that up here.
I don't think it's commercially available to.
Oh, that's right. It's for
your
labs.
Average person. It's really commercially available to labs.
That's right. Yeah, that's FDA just gave it an EUA here, so.
Because I've been thinking, that I know people who've been vaccinated and don't show up positive on an antibody test. Okay. And so, that does happen. Okay.
Of
course, I don't know if those individuals are immune either.
Yeah.
Okay. They're pursuing T-cell opportunities.
Yes.
But it happens with all vaccines. I mean, I've had 11 measles vaccines and I have no antibodies.
Yeah. And I will just, to sort of oversimplify a little bit, I will also just let people know that they should, if they are getting such an antibody test, they should make sure they're getting an antibody test that tests for antibodies against spike.
Yep.
I have received some emails from people who were very worried about not making antibodies after being vaccinated. And it turns out they had used an antibody against nucleocapsid and not, or they looked for antibodies against nucleocapsid, which is not in the vaccine.
Yep.
So also make sure you're looking for antibodies against spike.
I think actually the most upsetting thing here is the highlighting of the economic disparity that goes up throughout the world.
Yeah.
And the fact that you can't trust the government or anybody outside your family in some countries. I think that's actually, I don't know, that I found way more disturbing.
It is.
Yep.
Yeah, the guy wants to take care of his mom. It's great.
Grandma,
Grandma,
his grandmother, and he's far away. But the fact is that there's no, we take it for granted that while we have a lot of problems, at the end of the day, we kind of get it together. Whereas it doesn't sound like that's what happens in a significant portion of the world.
Well, maybe he can get a second vaccine for her.
Yeah.
All right, one more.
But who knows when that's going to be?
Yeah, well.
Good luck.
Good luck.
Yes, good luck.
All right, last one. Frederick, dear TWiV hosts, having followed and received science and comfort during a difficult pandemic, I'm wondering if you can provide us with your view on the possible thrombocytopenia from AstraZeneca adeno5 virus vectored vaccine, as there are news reports discussing adenovirus type 5 thrombocytopenia, pegylation, heparin-induced thrombocytopenia. Wonder what the causes certain people have to such a reaction. That causes certain people to have such a reaction. Is a specific human genotype a factor? And Frederick provides a link to Science Norway, in which Norwegian experts say that the clots were caused by the vaccine, which, to my understanding, is no longer thought to be the case. This has been looked at by AstraZeneca, and they've decided that these were events that would have happened anyway. They're at a lower frequency than you would expect in the general population. So I think you have to be careful. When you immunize many, many people, other things happen. And I think this is the case. What do we think? Any other thoughts on that?
I've given this some thought, and I had some conversation with some vaccine safety experts this morning about this. It is important that the European Medicines Agency, which is the Europe's equivalent of the FDA, has said that this is the benefits outweigh the risks. Okay, so don't stop using a vaccine.
Right.
I think there's two issues here. First of all, as you already said, Vincent, the incidence of blood clots in people who've had the vaccine is not higher than in the general population. And in some populations, as I understand it, it's even lower. So you could make the idea that the vaccine actually protects against it, but that's being mildly facetious. At any rate, so from that point of view, the risk of getting COVID far exceeds the risk of getting any sort of clotting disorders. That having been said, there are those who are still in the industry taking this seriously, okay, and investigating further to see if there could be any association between the vaccine and these disorders, however rare. Okay. And a number of different hypotheses have been put forward, but there's no evidence yet. And people are looking at all the things that you're talking about. Okay. Previous infection, genotype, vaccine lots, all different sorts of things. So this is being investigated further, but for now, the data are that the risk is so low. We're talking about less than 30 cases out of 20 million people, something like that. And I think one or two deaths.
Yeah.
Okay. So there is no real reason for concern statistically, but they are looking into it because it's an important issue to try and understand if it in fact exists.
And we should, the European, as you said, Medicines Agency has said this is not associated with, but it's also, there have been 11 million doses used in the UK and none of these.
It does seem, it's almost as if there's some sort of regional thing as well.
Yeah.
It's that from what I've seen, but I think of, ads for drugs that you see on TV where the list of possible side effects is longer than the ad. Mm-hmm. Okay. There are risks associated with everything we do.
Yeah.
Okay. And a lot of these risks don't show up until something is applied on a very, very large scale because they're very rare.
Yeah.
But just because there is a very low probability, and I'm not saying that's the case in this case, it's being investigated further, but the probability is so low that the risk is so much smaller. The risk is so much greater than getting a disease than any possible risk of the vaccine that it's crazy not to get vaccinated. Okay.
So there are a couple of things going on here that trip the alarm bells. First of all, clots are very common. This is something that, every day people are throwing clots and ending up, in the hospital or at least the doctor's office because of it. The concern here is not clots per se. It's that what people were post-vaccine, they were presenting with what seemed like a very unusual, very distinctive clot syndrome. And it's a whole pattern of symptoms. And I just pasted a link to a news article in Science that goes into the more of the details on this. So it was unusual in the sense that, wow, you don't see a lot of this, and they just got vaccinated. And so that's why many countries put a pause on this initially. They said, okay, maybe this is something. And yes, it's very few people, but this is a case where if people lose confidence in these vaccines, you're toast. So there's that too. Yeah. And we want to be ultra safe with this. So they paused it, looked at the data. Currently, the thinking is that this is probably not vaccine related. If it is exceptionally rare. And so the benefits of the vaccine still outweigh the risks, but it's something that deserves further investigation. And so that's what's going on right now.
And in this vaccine in particular, it's really important that we maintain trust in the vaccine.
Yes.
Because this is one that's inexpensive. It doesn't have a lot of the storage problems and is being distributed widely. And I feel for the people making the calls on this, because on the one hand, you don't want to ignore an alarm that somebody raises. On the other hand, you don't want to go off the other end as well and raise too many alarms yourself. Okay. It's sort of damned if you do, damned if you don't.
Yeah.
But the bottom line is that COVID is a bad disease and this prevents it. Okay.
Yeah. And just to reiterate, this is only the AstraZeneca vaccine. That is the only one of the COVID-19 vaccines that has shown this and that is being discussed. And that's in use in Europe. It's in use in quite a few countries. Is not in use in the U.S. And at this point, we're going to be awash in vaccine and probably aren't going to get this one at all. But it's being delivered to millions of people.
I have a suspicion that people are already wary of this vaccine, unfortunately, because of this, right?
Yeah.
And people are not going to want to take it, which is really too bad.
Well, there are this vaccine's had a rough road. I mean, it was really the earliest contender in many respects, and there was a lot of optimism around it.
Yeah.
They had a dosing issue in the clinical trial that, it turned out it worked better at half dose. Yeah. And the reason they found that out was because they accidentally gave some people half dose. But so we got the data on that. The phase 3 looked pretty good. The efficacy on it is maybe not quite as high as the ridiculously high efficacy of the mRNA vaccines that we've seen.
It was paused, I believe. Mm-hmm. It was paused because people had Guillain-Barré or some kind of.
Yeah, there was an earlier concern with it on that, and then that turned out not to be such a concern, and now there's this happening. I think it's a good vaccine. If that's the one that had been offered to me, I would have gotten it. I don't think that there's a serious indication that there's a problem, but it's had like every little thing happening in its development pathway.
All right, let's do some picks of the week. Brianne, what do we have this week?
So I have a TikTok video that went around social media a while ago. I hadn't gotten a chance to watch it, and I finally watched it and thought it was hilarious. It's a little video all about how mRNA vaccines work, and it's just great. And it made me laugh. And could I pick at the exact mechanism of antibodies working? Sure, but not that much. And it's just wonderful. And I actually think it would explain things to quite a few people who are unsure about these vaccines. So I like this little video so much.
Cool. TikTok.
Yeah, this is brilliantly done. I saw this passing around. Okay, yeah, this is really good.
Isn't it so funny?
And it's like a minute long. It's a TikTok video. It's the kind of thing that's very easy and fun to watch. And the approach is very creative and it's a very well done explanation.
Yes, the man who made it plays the mRNA, the ribosome, the immune system, the antibodies, and the virus at different times using dramatic voices.
Yes.
And it's just really funny.
Good. Alan, what do you have for us?
I have one that I actually, during the time when we weren't doing picks on TWiV because we were doing, Daniel Griffin on the show and everything, and it got too long, I started doing them on my blog. So this is one of the picks from that time. And this is a series on Netflix that is amazing. It's called Dark, and it's you can watch it either in the original German with subtitles, or you can watch it dubbed, whatever your preference. I watched it with the subtitles. It's 3 seasons. And unlike many of these Netflix series that, it's really good for a couple of seasons and then they get through the material that they had in their initial pitch, and the producers are saying, make more seasons, and they extend the concept and it doesn't do as well. This one is 3 seasons and it ends and it's over. And there's never going to be any kind of a sequel to this. And you will see why when you get to the end of season 3. It's a mystery. It's set in the fictional town of Winden, Germany, and it's set in several different time periods. This won't be too much of a spoiler. There's time travel involved. And it is just so good and so well put together. I was totally blown away by it. I really, really recommend it to everybody. And you don't have to keep track of everything. Don't worry. It will all be resolved in the end.
Good. I'm always looking for a series to watch.
Yeah.
I'll have a look at this.
I think it's always good to hear the original voices, right? And have subtitles on whatever, you know.
That's what I like about the subtitles.
Yeah, now you can pick that. When you get a dubbing is always so artificial, right?
Yeah.
Also, there are lines in this that are just so good in German, you know.
It's probably lost on me, but
I don't speak a word of German, but it just sounds so good.
that was good. All right.
Yeah.
Rich, what do you have?
So last episode, Dixon picked a YouTube video, I think it was, that was a series of little science experiment vignettes.
Right.
And following on that, I received a message from a listener named Volker. So this is kind of a listener pick that I'm copying here to a Science Everyday video. We've picked videos from Science Everyday YouTube channel, or Smarter Everyday, sorry, YouTube channel before, but it expands on one of the things that Vincent, in Dixon's pick, which is a reversible laminar flow, where you have a cylinder inside another cylinder with a viscous liquid in between. In this case, it's corn syrup. And you spike that with colored dyes, and you turn the internal cylinder and they smear out. And it turns out that you can turn the cylinder the opposite direction the same amount and reassemble the original spots. And so this is a drawn out version of the same thing with appropriate explanations. And of course, the guy who does it is like totally excited the whole time. So it's pretty good.
Cool.
It's fun.
Volker is the same one who I quoted last time as telling me that there's no such thing as cold weather, just inappropriate clothing.
Mm-hmm. Cool. Amy, what do you have for us?
So I'm not doing science this week, or at least not directly the science that we normally do. So one of my favorite sculptors is on his first solo exhibition since like the 60s is at MoMA, or starting at MoMA. It's Calder. And if anybody knows me, they know like that Vincent has the wall of polio, but I've always said that when I get in office, I wanted a Calder mobile of different viruses hanging above my desk. And so this is like a retrospective of his entire career, from when he did wood and metal all the way up to his intricate balancing of the mobiles and very large sculptures that you see at like Storm King.
Cool.
And stuff.
Excellent.
Awesome.
Instead of viruses, you should just have their genetic material, because they'd be mobile elements.
Yeah.
Cool.
No, I don't want retro transposons and transposition.
So are you gonna build your own mobile? Are you gonna have someone build it one day?
No, I'm gonna build my own, but somebody's else is gonna 3D print the virus.
Yeah.
Oh,
Maybe. So my friend's husband or boyfriend has a 3D printer,
I see.
and David is very handy. So we might try and convince David to 3D print some of the viruses, and he's very into it. So.
Well, yesterday I did a talk to University of Puget Sound, where Mark Martin is, and he's a big 3D printer. And he told me he has a 5-foot diameter virus in his office.
Whoa.
Wow.
And I said,
well, I don't need one 5 feet in diameter
I
hanging
understand.
over my head.
You need small ones, but I want a big one, because I want it in the new studio to hang behind me. And he said it could be arranged. Can you imagine shipping it from Puget Sound? I think that's a bit. Might have to put it in pieces, and I put them together.
You'd have to put it on a ship and worry about pirates stealing it.
Pirates. Hey, did that thing get to where it was going to go, Brienne? Do you know that ship?
I don't know. I think that one of the things that they were doing was keeping secret when it traveled.
The date. Yeah.
Because they didn't want the pirates to find out.
Right.
So
And it takes a while for ships to get places.
Yes.
So I wouldn't expect that to be real quick.
But why at 5 feet would you hang it and not have it like as?
Because he's going to have a 20-foot ceiling in the studio.
No, I picked out the studio, Alan.
Oh, okay.
The studio is my vision.
It's about the same height as this office, right?
No. So I would think it should be like a floor sculpture. And then you might actually want it a little bit bigger than 5 feet.
It's a good idea.
And have it as a floor sculpture.
Yeah.
We'll have to discuss, since this is the first I'm hearing of it. And my real estate agent hat is being replaced by interior designer.
I see.
So,
okay. Yeah, well, I just mentioned.
What you need is half of a capsid that can be built into a Dr. Evil style chair.
Wow, that would be fun.
Like an egg chair.
Yeah.
Like an egg chair.
And you can sit there inside a viral capsid.
That's a good idea. And it might not be so comfortable though, right? You have to pad it.
Well, no, you'd have to have an actual chair seat inside.
Yeah.
No, why wouldn't you just make the chair, the upholstery of the chair, like VP3 and VP?
Right.
No, VP3 and VP4 and have all the, that if you cut open a half capsid of polio, then you would have all of the VP, and then it would just be the upholstery of the chair.
So
how does that work for you, Alan?
Sounds good.
I mentioned
this is why I have the interior design.
I mentioned some time ago. I said, Amy, I want to have a studio with actually Daniel Griffin and I are sharing an office/studio downtown, and the next thing I know, she said we have an appointment to go see some space. So. Here we go. It is on 8th Avenue, just 2 blocks north of Penn Station, which is very cool. And Daniel will have his...
Is that like Hell's Kitchen?
No, it's the old garment district. So all of the old, and it's / the old garment district / the old music production studios. They've all moved out. There's no cutting of clothing anymore in New York City. And the music industry left in the 90s.
Wow.
And stuff. And so they're repurposing these large white square rooms.
Well, I'm excited to take the train and come visit.
Yes, you should come for sure, anytime, because it's right there. You get out of Penn Station and boom, 2 blocks. And for sure. And eventually I'll move the wall of polio down there as well, because I'm not going to be here forever, but until then. All right, my pick is a website called Ripple Training, which is a place which will teach you how to use video editing software. If you want to learn the major packages like Final Cut Pro or DaVinci Resolve, or Motion, this is the best. You have to pay for it. It's not cheap, but it is the best tutorials you can find if you want to. And this is what I do to learn how to do things. They also have a YouTube channel, which is free, and you can watch short videos about how to do things. But if you're interested in video editing, this is the place, Ripple Training. It's really the best.
Cool.
And I'm sure not many people will be interested, but in case you're interested in where I learn how to do stuff, that's one of the places. And we have 2 listener picks, one from Ruth. Hi, Vincent, and all the Fab team. It's about my first year anniversary of listening to TWiV, and thank you for keeping me and so many sane almost in a tough year. Just had my Oxford AstraZeneca vaccine and had the shivery shakes for 48 hours. Now okay. Just thought you'd like everyone to know that 100 million-year-old dormant microbes tucked up in the deep seabed are now up and out of the seafloor and doing fine and dandy, and they're eating and growing. I do hope they are held securely, as we can do without more microbial excitement for a decade or 2. And Ruth sends a story from Scientific American. 100 million-year-old seafloor sediment bacteria have been resuscitated, by Jennifer Fraser.
Wow. I want to sequence those guys.
Yes.
That would be
See
cool.
if there's any code evolution.
Yeah, that would be interesting. All right.
It's 100 million years.
There's nothing, huh?
And the spot that they drilled is Cthulhu's home.
H.P. Lovecraft, huh?
Yes.
And then we have a second pick from Iradis. Hi, TWiV team came across a highly interesting New York Times article regarding Linda Zoll's civil and environmental engineer turned science spy for the CIA from 1985 to 2013, and her program was called Medea. Yes, named after the stubborn Greek goddess. Maybe y'all have heard of her, but I had not until reading this piece. Very fascinating, and Iradis provides a link to the New York Times article by William Broad. Inside the CIA, she became a spy for planet Earth. She discloses how she toiled anonymously within the agency to help scientists intensify their studies of a changing planet. And I'd not heard of this.
This is cool.
Very interesting.
Oh, this is really neat.
Neat stuff. Thank you, Iradis. And that'll do it for TWiV 734. Show notes at microbe.tv. Send us your questions and comments to TWiV at microbe.tv. And if you like what we do, consider supporting us. Microbe.tv/contribute. Amy Rosenfeld's here at Columbia University doing plaque assays. Thank you, Amy, for joining us.
My pleasure. The plaque assays are calling.
I know you're a fan of Susan, so thanks for joining us.
I think Susan's great. I've always enjoyed talking to her. I spent a lot of time with her at ASG in Minneapolis. And I think she's a plethora of knowledge that is not really tapped into.
Yeah, I agree.
And and Alan, you will relate to this when you are trained in the Racaniello lab. Your idea of how to do research is you go to the old papers, you read them, you do experiments to where your system no longer fits into that model, and that's what you investigate. And that's what, and you read a ton of different things that are related, like all neurotropic viruses, we work on. So I don't just read the polio literature, I read Susan's literature and other stuff to understand how EV68 works. So yeah I really enjoy Susan. Yeah, I think she's great. Super intelligent.
Thank you, Amy.
Yeah. Thank you.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Fair enough. Always a good time.
Alan Dove's at alandove.com. Alan Dove on Twitter. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.